All right. I think we can start, can
Sure.
we? All right. So it's my great pleasure actually to have Vedran Miletić today as a speaker for these Friday seminars, and they've been quite a success. I've been enjoying them, and I hope I'm sure I will enjoy these as well. So a little bit of info about Vedran. Vedran is not a chemist, as far as I know. He's a computer scientist, and he has been involved into the development of several software. Some of them are open source, and he will talk a little bit about open source, I believe, today. He has contributed to science codes like CP2K for quantum chemistry calculations, as well as GROMACS for molecular dynamics simulations. It's one of the most used molecular dynamics engines. And well, he has been contributed quite a bit. I can see more than 70 contributions on GitHub just this year, 60 last year, and so on. And today we'll be talking about an idea that we had back in time of creating a web server that would perform docking studies, let's say, and would be way more accessible than whatever docking tool is nowadays. So I'm actually, looking forward to your talk, which is happening from Croatia, actually. And this is why we are having this meeting at 9:00. So thank you very much for giving the talk today, and I'm looking forward to it. The floor is yours.
Thanks, Dr. Mercadante. I'm very glad to be here. Thanks, everyone, for joining. I will, as Doctor said, talk about running RxDock on web servers in the cloud or in the university. So let's start straight away with a simple quote. So Francis Crick said that his own prejudices are exactly the opposite of functionalists. If you want to understand function, study structure. And he actually believed that structure determines the function of biomolecules. As we can see, his ideas actually have proven to be true. So what we are going to be talking is structure-based drug design, thanks, among others, to his work. What we want to do when we study potential new drugs is high-throughput virtual screening. If you want to analyze just one ligand on one target, let's say you have a complete MD analysis like molecular dynamics, as Dr. Mercadante said, with solvation, etc., and also do ADMET analysis like toxicity, Lipinski rules, etc., you need at least 1 week just for one molecule and one target. And that's assuming that you have a lab ready that will do this work. So that's not just, you have to first prepare your students, etc. You have everything ready. Everybody knows what they are doing. You need at least 1 week for all these studies. And let's say you have an average screening library of 100,000 compounds. So 1 week per compound on just one target node. And that means 100,000 weeks or almost 2,000 years. And the question is, do we have 2,000 years for initial screening? And I would say that we don't, but Dr. Mercadante's great-great-great-great-great-grandfather, Caesar, could have started the screening in his time and he would probably be done by now. But the rest of us don't really have 2,000 years, so let's see if we can do something else. The way these things are done, usually in reality, is that you do high throughput screening to pre-filter the compounds. Let's say in this picture it says like thousands of compounds, and then we filter the compounds and we have hit to lead part where we have not thousands anymore or tens of thousands, we have hundreds of compounds. Then we optimize the lead where we work with dozens of compounds, and then we seek the candidate molecules, which are 1 to 3 compounds that are more interesting than that. And then when we move to preclinical and clinical phases, we work with less and less compounds, maybe 1, maybe 2, we decide then. But anyhow, we can see this funnel where tens of thousands of compounds become thousands of compounds, hundreds, and then dozens, and then just 1 to 3 compounds for further optimization and testing. The reason for this is not only the fact that each of these stages lasts longer, it's also the cost. As you can see, computer modeling is much cheaper than human clinical trials. And these are the costs from 10 years ago. These are the last that I could find in the paper. And note that computer modeling costs actually decreased probably by 2 or 3 times, maybe even more. And human clinical trial costs increase. So it's not like $500 million, it's $1 billion right now, US dollars. So it's $1 billion right now per one compound. So these costs tend to increase, except the computer costs, which tend to go down. So if you look at state-of-the-art molecular docking, which can help us to filter these tens of thousands or hundreds of thousands of compounds down to hundreds or let's say tens of compounds, we have a few categories of software that exists in the wild. The first category is web servers. We can see an example on the right, which is called RxDock. It's just one of the web servers. We have commercial software like Gold or Glide or many others, and we have open source software. And these are the rough categories. You can divide them, even finer, as we will see very soon. When you start, you usually first find the open source software like Autodoc, like Seed, like RDoc and RxDock that I'll be talking about. And these are software where you can get the source and you can run them as you like. There are another open source projects which are more like a proof of concept that have very new methods and that they tend to prove that these methods work better in certain special cases, but are not really intended for general usage. More like a prototype of a method that can later be assimilated into other docking software when these methods prove to be working very well. Another category of software that you might be familiar with are docking web servers such as Galaxy, which work on the web. You have to prepare your files, then you find the web server online, then you upload your molecules, and then they do these things. They do their magic after 2 days, after 1 day, after 6 hours, whatever their time is, you get the results out. You have a number of solutions here. Some of them are commercial, some of them are demonstration that open source software works, some of them are academic, and there is of course a number of differences between them. But regardless of how you look at them, there are a number of concerns about all these web servers. The first concern is privacy. So you are studying some interesting proteins that, unlike COVID, not everybody in the world is studying, obviously. And you don't want everybody to know, at least everybody who has access to the files that users upload on the server, so the server developers, you don't want anybody else in the world to know what you are working on. And especially if your small molecules are interesting and you want to potentially patent them at a later date, you absolutely do not want them to be visible to anybody. And server operator might be somebody who understands enough chemistry to get an idea of what things could be done independently of your results and then patent the molecule far before you do it. So, not to mention that, you also get the accompanying results for the molecule. So if you have like 1,000 jobs and only 3 jobs actually get some interesting results, this server operator can actually see your results even before you see them. And aside from, potentially peaking server operator, you have the whole world of hackers. What if the hackers actually hack these public servers, which are public, so, they are visible to everybody in the world. So what if somebody anonymous from some country you have maybe even never heard of hacks the server, steals the data for everybody, and in particular your data? Your results get exposed to the hackers. They might want to sell them to pharma industry or to another university or to anybody. So what would be great if you could get these servers, like simple to use, but actually being in-house inside the university, accessible only to the university staff and not everybody else in the world. So they are not widely visible publicly, just internally for the university users. That would obviously solve the issue. And finally, there is another category of commercial docking software, where the most prominent software is Schrödinger Glide, quite famous one. There are other solutions you might have heard of Gold, maybe others. And all these software are very nice, but they are very costly. Their license is very expensive, and most of them or many of them have yearly license. So every year you have to pay again, and or if you want to upgrade to a newer version, you also have to pay again. So depending on which solution you choose, some are obviously cheaper, some are obviously more expensive, but they're all expensive compared to both the docking servers and open source software, obviously. Additionally, they also have no standardization. So most of them use proprietary file formats, which are usually not readable by other software. So you cannot really independently verify their results with other software. You cannot compare the results. And this is a bit tricky. So when you want to reproduce, you need to have the same software to open the same workspace, to rerun it, and to see how the results look. There is also a high entry barrier that I mentioned, so license costs are usually prohibitive, and you have to train the users because most universities, unlike commercial companies, don't really have the money to pay for these licenses, so students don't really come with skills to use these commercial software. And if you want a new feature, if you want to try something different, if you want a custom scoring function, if you want to try some different physics, etc., if you want to integrate them with other software, you're actually limited in the way that you can extend them. So you would usually ask the vendor to implement this interesting new method you want to see. You cannot really do this yourself because the license is proprietary and the software allows only the vendor to extend it and not everybody who actually wants to do it or everybody who is using it. With open source software, you don't have that problem. So there's another category of software that is kind of popular in the last few decades, and that is academic software that tends to be usually less expensive than commercial software, sometimes freely available, not often, especially not for commercial use. So academic software is freely usable for academics, or let's say lower cost for academics, but not freely usable, or let's say higher cost for the industry. It also has the same problem with limited adaptability. It's not open source, so you cannot extend it as you like. And there is an additional problem that commercial software doesn't have, that it depends on academic pursuits, because the amount of money that it gets is too low to actually pay the software developers as professional software developers. Usually rely on PhD students and postdocs to develop the software. So depending on how good are professors in obtaining funding, and depending on how are the funding calls written, it might or might not survive in 10 years or 20 years or even 5 years. This is not guaranteed. Usually commercial software gets more money, and the only thing they need to do is to hire software developers to develop it. And unlike academic software and commercial software, open source software is freely available to anybody. It has full adaptability, so anybody can develop it as they see fit. And industry and academia and volunteers, anybody can use it as they like, for any purpose as they see fit. They just need to respect the changes. So if they change something, they have to open source their changes. That is called like playing fair. So everybody shares the code with everybody else. In particular, the software that we are working on is RxDock, which started in 1998 at a company called RiboTargets, later renamed to Vernalis. Company started as only a computer software company in domain of computational chemistry, and later expanded to also in vitro methods. So they still do in silico, but they also do in vitro these days. What they developed was RiboDock, the commercial high throughput virtual screening software. Later they renamed it to RxDock, and they gave it to University of York for academic licensing. Then the software also became cheaper and could be more easily obtained. And in 2012, they actually open sourced it and let University of Barcelona develop it further. And unfortunately, due to probably funding constraints and other interests, development mostly stopped in 2014. But unlike academic software, which if development stopped couldn't be resurrected, in this case, this software was open source, so it could be resurrected, and it was resurrected by us as RxDock, which I will talk about very soon. First, let me tell you that Vernalis has a platform that they offer to other companies. And one of the companies that actually bought their drug development platform is Ligand, that is available on ligand.com domain. And Investopedia recently listed it as one of the top value stocks at the moment. So they are expected to grow very rapidly. And they're like the best stock to invest, like among Galapagos Group, where my co-founder is actually from. He formerly worked at Galapagos Group. So these biotech stocks which use Vernalis design platform are actually doing very well. And what we did, we took this software that was formerly developed by Vernalis. We actually extended it by inside of our company and decided to continue releasing it as open source, because what made it possible for us to do it was that it was in fact open source in the beginning, and our first goal was to get it running on modern PCs. So when we found the code, it didn't really run on modern PCs. It was a few years old. So first we decided that we wanted to get it running on, your average PC, your average laptop, because, we want to have users readily use it, very easy to start. Primarily we use Linux and sometimes we use FreeBSD. But we also support macOS, we also support Windows. Dr. Mercadante is a prominent user of macOS, so he would be very angry if we didn't support macOS properly. We also support Windows, obviously, and other operating systems which are less known. Hopefully, in the future, we will reunite with RxDock, because we have been noticed by the original authors, Peter Schmidtke from Disk Engine, a French company, who said that it would be interesting if we could merge our work back into the original project. Since original project has been dormant for 5 years, maybe we could, join the forces and not rebrand. We will see how that goes. We are open to these ideas. We had a meeting like 2 weeks ago or so, and we'll see how whether something develops or not. If not, we are having a separate software. If yes, then we will be calling our software RxDock. Either way, we will continue to develop it, and that's the beauty that open source actually allows us. Other things that we developed after we obviously made it support the modern operating systems. We first worked on the documentation. You can find it on rxdock.org. I will not be opening it right now to interrupt the presentation, because, sharing screen can be tricky to get right. So clean, structured, easily searchable, both for online viewing and for printing. As I said, on rxdock.org, you can see it later how it looks. That's the first step, because software starts and software dies with its documentation. Bad documentation, good software, nobody will know how to use it. Good documentation, bad software, people will at least try to get it working as the documentation suggests. So good documentation is absolutely essential to get users started on the software at all. What we also developed was a frequently requested feature where RxDock or RxDock measures the performance and estimates how much will it take to run the rest of your library that has 1 million compounds. So it will finish on Thursday at 2 in the morning. So you know how long will this take. Do you want to leave the computer running on for 3 weeks? You might want to, you might not want to. And there was another issue, especially with these long runs, that if it finds one broken molecule, you have to stop it, because the software runs forever or crashes. And in this case, we just skip the broken molecule. We don't let it run forever. We don't let it crash. We just check if the molecule is broken. That's fine. Okay, we'll just skip this molecule. And then at the end, we'll tell you, you had this number of molecules that are skipped. This was all from feedback from the users, because we listen to user feedback a lot. And we want to actually develop features that users care about. I mean, we have our own internal interests, but we also listen to the community, and we want to develop things that community wants. And at the moment, we are working on performance improvements that will have much faster sorting and filtering of results. So let's say you get a scoring of 1 million compounds, and you don't want to have 1 day of sorting of these molecules by score. So you would like to get this done in half an hour or maybe 15 minutes. So we managed to improve this by, let's say, a factor of 10 to 100 times, which is quite impressive, and we are quite proud of it. And after a bit more testing, this will be made public. We actually did some work on parallelization in cavity search phases and post-search phases. So this is also still internally being tested and developed. When this is ready, this will be pushed out. We are working on checkpointing, so if your run crashes, that it can resume where it stopped, not from the beginning. And hopefully we'll offer you the graphical user interface and more streamlined command line user interface. In case of graphical user interface, that would mean you run PyMol, and then you get one-click high-throughput docking. You just load your protein, you load the potential compounds that you see how they dock to the protein, and then just click, you get it all in PyMol. And the key feature that Dr. Mercadante actually mentioned in particular is RxDock web server, which we are developing at the moment. This is a mock-up. This is how the user interface might look finally, where you can use the web server, upload your protein, upload your ligand, choose the cavity mapping method, and then just let the server run docking on your ligand or ligands, or even if there are more 1 million ligands, you just let it run, and when the results are done, you just get a notification by email or any other way that the results are done and that you can download them. So this would be the most streamlined way to use it, and this is the target that we are hoping to have done in, let's say, 2 months. 2 months working prototype. We'll see. In any case, what we use in this case, the science behind these things is that RxDock actually finds the cavities by putting the spheres on your protein. So the method that we use is called two-sphere method. We try to find the spots, like the holes on the protein, the cavities on the protein, where the small sphere can fit, but the large sphere cannot fit. So that's how we discover that there are some cavities which might fit a ligand at this protein. This is the case where you don't know where the ligand binds to the protein. If you do know, then there is another method for cavity mapping, which is called the reference ligand method, where you find the volume around your ligand. So in this case, you actually have a ligand which you start looking how it looks, how it fits, and then you start guessing how does the cavity around this ligand look, and then you use this to dock all the other potential ligands that you had in your compound database that you used. Both of these methods are obviously supported via the web server. Both of these methods are supported via the command line, so they are offered to you as a method of choice for docking. There are many ideas for new features that we would like to develop over time, obviously. We would like to work with molecular descriptors and QSAR integration. There is some work already being done in this direction of molecular descriptors. We would like to have other solvation methods. We would like to have genetic algorithms that would automatically design the compounds. From a certain starting compound, you would get all these variations. We would like to have a streamlined statistical post-processing of docking results, so it doesn't have to be done manually. We would obviously like to support more variety in custom scoring functions. We would like to have pharmacophores and in general fragment-based docking, so you would dock fragments instead of the whole compounds. We would like to support an ensemble docking, which is quite popular in the last few years. We would like to integrate molecular dynamics, but this is like a far-fetched idea. We'll see how that goes. And obviously, we would like to use artificial intelligence and neural networks in docking, which would be hopefully giving us better results. And obviously, we are open to other ideas. This is just, some blue sky visions that what could be done with RxDock. These are just some ideas that we might, want to do in the near term or long term. That are not currently supported or not currently supported well enough, but we are open to other ideas from the users, which hopefully includes you in the future. And what our use case is, and let's say what our competition is doing, is this. So in 2012, Schrödinger actually ran for $5,000 per hour on Amazon AWS a docking run that had 50,000 computer cores and was looking for a certain drug that would target certain protein interesting protein in development of cancer. So that's like $15,000 for 3 hours. Let's see whether that's the whole cost. Well, it unfortunately isn't the whole cost, because in addition to the paying for the compute hours, you also have to pay for the licensing. As you can see, licensing is, let's say, 5,000 to 10,000 United States dollars per year, either for the web server or for the software that you can use on site and in the web server. And I'm not sure that this license would actually give you 10,000 core or 50,000 cores or 100,000 cores in the cloud. Maybe it give you like 500 cores or 5,000 cores, maybe not on the scale that they were running it in the cloud. But let's say it actually gives you this. You can see that these costs are not only very expensive already, they're rising over time. So cheap software, which is alternative to Schrödinger, was around $3,000 back in 2013. You can see that Schrödinger listed a price of $7,500 per year in 2014, and $5,000 maybe for countries that are not as rich as the New Zealand, Australia, or Germany, or USA. Countries like India, which are poorer countries, obviously they cannot charge as much as they would charge us. But these costs, regardless of where you are, keep going up. So even in University of Nairobi, Esther mentions that there is a cost of $10,000 per year for the license, and Bartosz says that it's north of $10,000 per year. So it's, basic starting price for you, what you want is $10,000. And that's limited license. You don't have infinite number, of course. So let's say $10,000 is some kind of license that we might be looking at. So what can RxDock do for you for a similar price or on a similar scale? If we were to take 50,000 core supercomputer in the cloud or at the university, assuming you have such a big machine, we would pay for it, let's say, 250 United States dollars per hour. So that's a lot cheaper, maybe 20 times cheaper than what we had a few years ago. That's not including the license costs. If we include the license costs, that's like 100 times cheaper, I think. So we are having much, much cheaper configuration already just by using the software that is open source on more modern hardware. If we have a database of 21 million synthetic compounds, in 3 hours we would get 10 to 20 poses per each ligand, so 10 to 20 dockings per ligand. Some people would say that's not precise enough. Some people would say that that's a too rough selection, that you might want to have 100 or 200. In that case, you would still have it much, much cheaper than you would get it with paying for the license and paying for the compute servers before. So open source actually would save you a lot of money in this case, and you would get dockings as well as before. So what we are doing here is that we are pushing this RxDock in a few directions, and we are in contact with several universities and research companies that we are collaborating with in this area. The company I already mentioned was the company where Peter Schmidtke works, which is Disk Engine, where we are actually interested in evolving the RDoc that they actually use internally in their cloud services. There is also University of Barcelona, where we are in contact with one of the original authors, Javier Baril, who is also interested in further development. And we are in contact with University of Ljubljana, who is actually using RxDock on a mass scale for COVID, for targeting COVID with various molecules. They have this Citizen and Cyan project where they use many people's laptops or even mobile phones, why not, desktop computers and other machines to test different compounds that might target proteins that are in coronavirus. So this is kind of folding at home done with docking, and they try to use a commercial software. But the license costs were very prohibitive, especially on this scale with 1 million users or 100,000 users or 5 million users, however far they get. And they actually contacted us and asked if they can use RxDock. We were obviously happy to give them RxDock for free. We did some customizations in particular for them. They have a nice user interface that actually does only their docking. So we had a really nice collaboration, and I hope their project actually succeeds in finding some interesting drugs that might be worth studying later in the lab. Aside from these studies, we already have a number of studies that were successful thanks to docking. I'll just quickly go over these, where they found HIV binding site and HIV drugs thanks to docking. If they were just doing this in vitro, they wouldn't guess this molecule, or they would take much, much longer time to get this molecule. In this particular case, what they did was first some MD to relax the protein, and then they did docking with the relaxed protein. So these methods are obviously not limited to just molecular docking. You can also combine this with molecular dynamics in various ways. Then there is another story where you had the same molecule discovered independently using virtual screening and by traditional methods with high throughput screening in vitro. And you can see that in this case, if we are doing all these studies in vitro, we would have to do large in vitro studies, which are very expensive in terms of manpower and in terms of all these kits that you have to buy, obviously. But if you do in silico, then you filter these molecules much, much quicker. So you can see that there are two papers that are published roughly at the same time. But people who did this in silico were much, much quicker to come to the same results. And not to mention that they spent much less money. And there's, so this was like 10 years ago, 20 years ago. These days, like in the last decade, you don't start with thousands or tens of thousands of molecules. You start with databases, as you can see on the right on this picture, with 2 million molecules initially. And if you start with 2 million molecules, you have to first filter them. And in this case, they filtered them according to the warhead, because they were designing covalent inhibitors. They knew how the binding part of the inhibitor had to look. And they filtered the molecules from all these databases that had the interesting warhead. So that they had the warhead that actually could fit and make a covalent bond with the protein. When they found these 1,247 compounds that were of interest, they actually expanded with stereoisomers such as enantiomers, and they got to the library of 2,100 compounds roughly. And then they went through the funnel that they were shown in the first few slides, where they reduced and reduced and reduced. And finally, they had the three compounds that are in vitro lead compounds for further development that can go through all the rest of the stages that I mentioned. So by the time they got to in vitro, they had already 21 compounds. Start with 2 million compounds, get down to 21 compounds just with in silico work, which is quite a simplification, especially considering that these days you can start with 5 million compounds, and these numbers of compounds that are commercially available to you only keep rising. In this study, they actually targeted GAB1, and they started with 5 million compounds. Just with molecular docking, they reduced this to 10,000 compounds. Then they filtered it using pharmacophores. They reduced it to 2,800 compounds approximately, and then they visually inspected them, etc., etc. Finally, for experimental in vitro validation, they had 20 compounds, which is a feasible number, obviously much more feasible than 5 million compounds. Let's note that in order to get to a part where you can do molecular docking, you obviously have to do preparation. So you have to do usually homology modeling. You have to refine the homology models you do. You have to see how the protein behaves with MD, etc., etc. So you usually cannot just start with rough protein that you get on PDB, etc. So you have to refine the protein with a little MD or with maybe some homology modeling if you don't know the entire structure. So before docking, there is quite a bit of work. But after you get into docking, the funnel starts that I said. So docking is not the method to end it all. Obviously, you can combine it with other computational methods. And finally, one very recent paper from 2019, where they use docking to get from the target selection to the biological evaluation in just a month and a half. So this is the scale that research can be done these days. So in 6 weeks, you get from the target selection to the part where you can get into further in vitro studies. Some initial in vitro studies were already done. So this is possible thanks to very good software that we have these days. As you can see, they selected databases of compounds. They did some pharmacophore search. They later even do synthesis, etc., etc. So this can happen very quickly if you have the pipeline prepared and with software that you know. So this is not something that needs to take a year these days, obviously. And as you can see here, these compounds found via in vitro and in silico studies actually match quite well. The picture B in the middle is an example how does root mean square deviation look in all these compounds. As you can see, the first two match almost exactly, and the third one is quite good in part, but not entirely matching. But these results are like very, very good, and with a small MD that you would do after docking on 6 compounds or 3 compounds, you would get the compounds positioned exactly, like very, very quickly. So what we are offering you is the web server that I mentioned that can help you with research such as this. This web server would be offered to you on a pay-as-you-go model that would not ask you to pay for the license, but instead would charge you per the hour of usage. This charge would cover hardware costs, so the costs that were paid for the hardware can be recovered by the university. Electricity that is used by the server, obviously also for the university, maintenance, which is done by us, and support, which is done by us. RxDock is and will always remain open source, obviously, so we will not be charging for the software. You can always install our software on your any desktop machine, any laptop, anything, and use it without any limitations. This is just for the web server, which offers a streamlined, nice user interface, which is running on separate hardware in the same university, which is your private hardware inside the university, isn't accessible on the outside to non-university people. And as I said, these costs would actually help the university recover this initial investment and would help us maintain the software for you. Of course, in this case, you would get new features occasionally if they were developed by the community or if they were developed by us for our own needs. New features will be coming over time, every 6 months, every year. There will be something new. But if you have a feature that you want to see, if you have something that you are interested, if something here caught your attention, that would be great. We can always offer you user-requested feature development as a service. In this case, it can be a contract between the RxTx and the university, or it can be a joint application for public funding, which we also like to do and sometimes do with our other collaborators. So either way, we are open to hearing from you, and I would like to end with this slide, and thank you for listening, and I would open the floor for questions.